This video is Chapter 6 Notes, Part 2, all about glycolysis and the Krebs cycle. We left off with ATP. ATP is an energy-storing molecule. It is what cells use to do specific jobs. ATP stores energy in phosphate bonds, such that when a phosphate is removed from ATP and that phosphate attaches to a protein, it changes the shape of the protein, allowing it to do a job that it couldn't do before. Also, that phosphate carries with it a small amount of energy, giving that protein energy. The goal of cellular respiration is to make molecules of ATP. To do that, a cell uses sugar or glucose, and it breaks that glucose apart to make ATP. Cellular respiration occurs in 3 steps. The whole goal is to make ATP. In the process, some water is made as waste, and also carbon dioxide is a garbage waste product that gets released by the cells. In order to break apart the glucose molecule and get the most amount of ATP made, the cell will use some oxygen. So the whole purpose of breathing is to take in oxygen so that your cells can break apart glucose, and then you expel that carbon dioxide garbage as waste. There's 3 steps to cellular respiration: glycolysis, the Krebs cycle, and oxidative phosphorylation. This video will focus mainly on the first 2 with a quick little intro to what oxidative phosphorylation is. This word oxidative might be familiar to you, but it might not. That is a chemistry term relating to oxidation. To help me remember what that word means, there's an acronym, Leo says grr. Like Leo the lion is growling. Leo the lion says grr. And just thinking of that helps me to remember these terms. Leo is an acronym that represents losing electrons. As you remember from chemistry, electrons are energetic. They are flying around the nucleus, orbiting around the nucleus. So electrons have energy. During cellular respiration, molecules pass electrons between them, and that passing of electrons passes energy between the molecule. When a molecule loses electrons, it is therefore losing energy, and chemists say that it is oxidized. That molecule, when it loses an electron, undergoes a process called oxidation, the loss of an electron, the loss of energy. Grr reminds me of the opposite. When a molecule gains electrons, it is gaining energy. It is then said to be reduced. The molecule is reduced, and that is the process of reduction. So during cellular respiration, you'll hear those words oxidation and reduction as electrons are passed between molecules to pass energy, ultimately trying to make ATP. The first step of cellular respiration, there are 3. So the first step is glycolysis. Glyco comes from the word glucose, which is the sugar. Cells eat food, they get sugar from their environment, and lysis means to break. So during glycolysis, that sugar molecule will get broken down. The sugar is a ring, glucose, and essentially during glycolysis, it's going to get chopped in half to get two pieces, each of which is half of that sugar molecule. Glycolysis occurs in the cytoplasm of the cell. That's the liquid of the cell. All cells have cytoplasm. All cells do glycolysis. Little throwback to evolution. An evolutionary biologist would say, well, if all cells do the same exact process, I believe that that is strong evidence that all cells came from a common ancestor. The first initial cells on the planet 3.5 billion years ago did glycolysis to break down sugar to get energy. So therefore, it's likely that all cells today are related. Glycolysis does not use oxygen. So cells that live in very rough and tough environments, like those on the ocean floor, don't need oxygen to do glycolysis. Also, they do not need a mitochondria, so bacteria cells do glycolysis. During glycolysis, there's two main products, ATP. Yay, that's what we're hoping to make by breaking down sugar. The whole goal of cellular respiration is to make ATP, so that's good. And then we also make this new molecule called NADH, which you've probably never heard of before. But NADH is a high energy electron carrier. Meaning it acts as like a little shuttle bus to move electrons between one molecule and bring those electrons to another molecule. So NADH, I always think of it as a little shuttle bus. It's carrying electrons, it's carrying energy. NADH is made from a molecule called NAD, and NAD gains electrons. So that's GR, that's reduction, it gets reduced. And it's gaining those electrons from glucose. So glucose is our sugar molecule that's coming in, storing all of this energy. So we're going to make in glycolysis NADH, which is going to shuttle the electrons that were in the glucose over to a different molecule. During that process, since NAD is going to be gaining electrons, well, there must be a loser who's losing electrons, and that's my glucose molecule. So always when I have a molecule gain electrons, there has to be a loser. So my molecule that's losing electrons is the glucose. And then at the end of glycolysis, we will have a molecule called pyruvate. So now a bit more on glycolysis. I'm going to kind of map out what's going on in glycolysis. If you Google glycolysis and look it up on your own, you'll see a whole lot of fancy enzyme names and what all of those different enzymes do. For our class, first intro level biology class, we don't need to memorize all of these enzyme names or know exactly which hydrogens, oxygens, and carbons they're modifying. So I'm going to show you here in this video like an overview, and those are the details that you're expected to know, not every little intricate step. Okay, so to get us started, in glycolysis, the starting molecule is glucose. That molecule enters the cell, and then it is in the cytoplasm where there are a bunch of enzymes, meaning a bunch of proteins that will modify that glucose molecule. The initial steps of modifying that glucose are called the energy investment phase. And the reason that it's called an investment phase is because actually the cell needs to spend ATP to modify it. So the cell is not immediately getting more ATP, it's actually spending ATP that it already had in storage. So during that investment stage, 2 ATP are used. And what's going to be happening is they're used by enzymes to break that molecule in half. Then, continuing, more enzymes latch on to that glucose molecule, which is now broken in half. And those enzymes continue to modify, and that is what we call the energy payout phase. Meaning that now finally we are getting some energy out of those bonds in the glucose molecule. So 4 ATP are made. But since there was already 2 used, it's really only a gain of 2. Also, during that process, NAD becomes NADH. So NAD is a molecule that the cell has available in it, and that NAD is ready to act as a shuttle bus to grab some electrons and shuttle them elsewhere. So during the energy payout phase, we make 2 NADH. So we essentially have 2 NAD molecules that grab electrons and we make these little shuttle buses. At the conclusion of glycolysis, we have ended now with 2 pyruvates. These are half pieces of sugar. We have ended with 2 ATP, and we have ended with 2 NADH. These items are going to then be used in the future steps if the cell can. So there are certain conditions that would allow the cell to continue on through the other 2 steps, or certain conditions in which case the cell would stop essentially at glycolysis. So during glycolysis, as we just said, we are breaking down glucose to make 2 pyruvates. At that point, the cell will either undergo anaerobic or aerobic respiration. Anaerobic respiration will occur if there is no oxygen available or if there's no mitochondria. So if either of those conditions are true for the cell, it will be forced to go into anaerobic respiration. In anaerobic respiration, the cell does glycolysis as normal, and then it undergoes fermentation. You may have heard of like lactic acid fermentation. That's what occurs after strenuous exercise when you get like a burning, crampy type feeling in your muscle. That is because you are exercising too fast for the oxygen to reach those cells. So those cells were forced to undergo fermentation. During fermentation, no additional ATP is made. So all the cell gets from a whole molecule of glucose is 2 measly ATP. That's really not a lot. A glucose molecule can store maybe enough energy for about 30 to 38 ATP. Different sources say different exact numbers, but about 38-ish ATP. So making only 2 ATP from that energy actually wastes a lot of the potential that's in that glucose molecule. So fermentation is not really ideal. For a single-celled organism like bacteria, that doesn't need to do nearly as many reactions as your more advanced cells do. Those 2 measly ATP are just fine for the bacteria cell to survive on. But your cells have high energy needs because they're doing so many reactions simultaneously that your cells really could not survive long term only by doing glycolysis. So a little bit more on fermentation. So why does fermentation even occur? If we can't make any more ATP, then isn't the cell just done? It did glycolysis, and so it should be done. Well, the problem is that during glycolysis, we had that molecule called NAD, and it picked up electrons to make that molecule called NADH. And those electrons need to be picked off of the glucose in order to make the ATP. So if I run out of these NAD molecules, I would no longer be able to make any ATP. I need those NAD molecules available to be grabbing electrons so that I can ultimately make ATP. So at the end of glycolysis, I've got NADH. Well, I don't have a lot of NAD remaining because they were converted to NADHs. So the goal of fermentation is to return NADH to NAD. That way my shuttle bus is empty and it can go pick up new electrons, and it can continue to break down glucose molecules to make those 2 ATPs. So we need to return an empty shuttle bus to grab more electrons so glycolysis can continue. If I straight up just stopped at the conclusion of making the pyruvates, I would have a lot of NADH in the cell, and I would be able to break down no additional glucose and make no additional ATP. So that would be a problem. The cell would run out of energy. Like I mentioned, muscle cells do lactic acid fermentation. Bacteria cells also do fermentation and produce lactic acid. Yeast cells make ethanol through fermentation. But fermentation, like I said, is not the ideal pathway if you have oxygen and a mitochondria. Then you want to go to pathway number 2, which is the aerobic pathway. The aerobic pathway requires oxygen and it requires a mitochondria, so both need to be there. The benefit of the aerobic pathway is that we can make about 30-ish plus more ATP from that same glucose molecule. You will never see a situation where one pyruvate goes to aerobic and one goes to anaerobic. No. They're pretty much always going to go to aerobic if they can. And if not, then they'll default to anaerobic. So to conclude a little bit on the Krebs cycle, which is step number 2. Step number 2 is on the aerobic pathway, meaning it requires the mitochondria, and the cell will only do that if there's oxygen available. The Krebs cycle, though, does not directly use oxygen. All right, here we go. Krebs cycle should be a bit faster. So we've got pyruvate. That pyruvate was made during glycolysis. If glycolysis does not happen, Krebs cycle is not happening. We've got to use the end product of glycolysis in order for the Krebs cycle to start. Krebs cycle occurs in the mitochondria. The goal is to pull more energy, to pull more electrons off of that pyruvate molecule. Again, if you Google it, you'll see a lot of intricate enzymes and their pathways. We don't need to know that for our particular class. We just think about the big picture. The big picture is that the Krebs cycle is going to occur twice because there's 2 pyruvates, and the goal is to pull more electrons and pull more energy out of those pyruvates. During the Krebs cycle, we end up making 2 ATP. So we make some ATP, but not a lot. We make more of those electron carriers. We make a new electron carrier that we didn't see before, but it works the same as NADH. It's called FADH2. That's another electron carrier. And in this process of the Krebs cycle, we also make CO2, which is our waste garbage product. Still, we haven't used oxygen. And during this Krebs cycle, we're making a small amount of ATP, but the money maker of the Krebs cycle are these electron carriers. They will come into play in step number 3. At the conclusion of the Krebs cycle, we have made 2 ATP in the Krebs cycle, and we have made 2 ATP from glycolysis, giving us so far a net gain of 4 ATP total. Since the glucose molecule stores enough energy to make about 38 ATP, we've made only 4, so there's still a huge piece of the puzzle missing, and that is step number 3, which we'll hit in the next video. But lastly, before you close out here, one last thing. If you look up the Krebs cycle, you'll see that it also goes by another name. So I just wanted to add that in here. The Krebs cycle is also known as the citric acid cycle. So Krebs cycle is the same as the citric acid cycle.